Hello everyone, this is Dr. Vishal Trivedi from Department of Biosciences and Bioengineering, IIT Guwahati. So, what we are discussing in the course Cell and Molecular Biology, so far we have discussed about the organism or origin of life, we have discussed about the evolution, we have discussed about the cell growth and regulation, we have discussed about the apoptosis, necrosis, and then we also discussed about the autophagy. And in couple of previous lectures, previous modules, we have also discussed about the central dogma of molecular biology and immunology. So, in this particular module, I have taken few selected topics on which we are going to discuss. So, today we are going to discuss about the gene therapy. So, what is gene therapy? Gene therapy is the use of any collection of approaches for the treatment of human disease that depends on the treatment of DNA based genetic material into an individual. It may be a gene which is going to be introduced, it is going to be removed, or there will be change in the genetic material. So, use of genetic manipulations by the human for the therapy of human disease is a new and rapidly evolved field, and the purpose of gene therapy is that it should actually be able to correct the disease at the genetic level, right. So, this is been proposed, or this is being utilized for the treatment of cancers, hemophilia, cystic fibrosis, spinal muscular atrophy, ADA, SID, and many more, right. The global gene therapy market, which is valued at the 7.2 billion in 2023, now is being increased to 8.85 billion in 2024, and it has been projected that this value could be reached to as big as 36.65 billion by the year of 2032. Now, how the disease, why we need to do the gene therapy? So, if you get a disease, right, you are actually having the three different approaches to take care of the disease, right. You have the option of taking a medicine, right. So, you can be able to reduce the progression of that particular disease. You can actually be able to use the surgical procedure, so that you can be able to remove the diseased organs or the diseased tissue. But the gene therapy actually take care of the disease at the root cause, right. So, it actually cures the cells, and it cures the genetic problem what is associated with a particular cell, and that is how it does not require the supplemented with the any kind of medicines, or it does not require the surgical procedure. Now, this is very advantageous compared to that you are taking a medicine or going with the surgical procedure, because the gene therapy is going to work on the root cause of the genetic problem which is associated with a particular disease. So, as I said, the gene therapy is going to address the root cause, and root cause which is associated with the genetic alterations. It is going to be treated even for the parents, right, and it can actually be able to bring down to the changes into the offsprings, and it also can be treated the parents for their own disease as well, right. So, how the field of gene therapy is being evolved. So, it started with the year of 1960 to 1970, when the concept of gene therapy emerged, with a key paper in 1972 outlining its potential and the ethical considerations. So, people propose that if there are possibilities, if there are the advancement into the genetic manipulations, why not we actually alter the genes so that it can be cure the genetic diseases. Then the year of 1980, it is decided to focus on the fundamental research, particularly on using the viral vectors to deliver the gene, leading to the first trial proposal in 1989. Then in the year of 1990, the first successful gene therapy trial was conducted on the De Silva, who had the SCID, making a major breakthrough. But in the year of 1999, the field trials were suffered a setback with the death of the Jesse, and that which actually been failed because of the anaphylactic reactions, or I will say the site reactions to the viral vectors. Then in the year of 2010, the first commercially gene therapies were introduced, including the Glybera, in Europe, and the Luxturna for inherited retinal diseases. And these kind of the CAR T cell therapies are being tested till the 2017 actually, and then 2012 to present the development of the CRISPR-Cas9 mediated genetic modifications and genetic manipulations has revolutionized the field of gene therapy, enabling the precise editing of the genome. And then in the 2020, the first CRISPR-Cas based gene therapy was approved to treat the sickle cell anemia and the beta thalassemia, offering the functional cures for these diseases. Now, talking about the gene therapy, the basic principle of gene therapy is that it is actually going to take up the healthy genes and it is going to introduce that into a patient, right. So, imagine that a patient is suffering from a particular disease, and in that disease one of the gene is non-functional, or it is not been able to produce a particular enzyme, and then what we will do is we will take the healthy gene from a healthy individual, and then we will introduce that into a patient, and once you replace the defective gene with the healthy gene, you are actually going to produce the particular protein, and that is how you can be able to cure a particular disease. Now, talking about the approaches what you can use into the gene therapy. So, you can actually be able to use the multiple approaches. So, as far as the gene therapy type is concerned, we can have the two different types of gene therapies. We can have the somatic gene therapy, we can have the germline gene therapies. So, somatic gene therapies where the somatic cells are actually going to be treated, and it is not going to be go into the offspring. So, it is actually only going to treat the parents, or it is only going to treat the particular individual. Whereas, when you do the germline gene therapies, the germline therapies are actually going to done for the germ cells like the sperms and the ovum, and then it is actually go into the offsprings, and as a result of that it is actually going to cure the genetic disease. Apart from that, you can also do the genetic modifications. So, you can actually do the gene replacement, you can do the gene corrections, or you can actually do the gene augmentations. And as far as the transfer method is concerned, you can actually use the viral vectors, or you can actually use the non-viral transfers such as the physical method or liposome mediated methods and the chemical method. Now, talking about the first therapy, that is the somatic gene therapy. So, somatic cells are a component of the all cell except the germ cells. So, you know that the germ cells means the cells which are responsible for the production of the offspring such as the sperms and the ovum. So, apart from these cells, whatever the cells are present in an individual are actually falling into the category of the somatic cells such as liver, lungs, brain, and all other type of endothelial cells. So, these in this particular somatic gene therapy, you are targeting the modification of the gene of the somatic cells. So, you are actually going to cure the particular individual for different types of defects. It is non-inheritable, so it will not get into the offsprings, and the gene transfer via the viral vector or the liposome mediated vector or the direct insertions. The examples are the cystic fibrosis, adenosine deaminase, or the cancer. You can actually do the virus escape, or the disruption of the important gene is a drawback of this particular gene therapy. So, in this particular gene therapy, there is a problem of virus escape, that when you are injecting the virus for the delivery, there is an immune system which is actually going to act against the viruses, and that is how you are not going to get the desirable results. Taking an example, so what are the target sites for the gene therapy? So, you are actually going to have the target site like all the somatic cells such as endothelial cells, liver cells, muscle cells, and the lung cells. So, this is all given here, right. You can actually do the somatic gene therapy in endothelial cells, brain, liver, muscles, lungs, and the skin, right. So, and remember that these are not inheritable to the other offsprings. So, it is not going to only cure that particular individual. Talking about the examples, so how the gene therapy works. So, we have taken an example of the cancer. So, cancer is the largest second largest leading cause of death in humans, and cancer occur as a consequence of accumulation of genetic modifications which leads to the uncontrolled cell death. So, you can have the uncontrolled cell division, you can have the loss of apoptosis, you are going to have the angiogenesis. Because of that, the cancer is going to spread from one site to the another site. Then you can also have the metastasis, which actually helps the cancer cells to move from the one side to another side. Then you also going to have the rapid glycolysis, and you are also going to have the loss of contact inhibition, and because of that the cancer cells are actually going to grow in large number. Now, when you want to treat the cancers, you are actually going to take care of the different types of mutations which cancer cells are acquiring, and because of that they are actually going to acquire the uncontrolled cell division. So, what are the approaches you can use? So, you can actually treat the cells which are associated with the cancer. So, you can actually use the oncogene or the proto-oncogene such as the EGF, CRAS, ERK1, CF and CMYC and the HER2. You can also use the some of the genes which are tumor suppressor genes. So, the function of these genes is that they are actually going to suppress the proliferation of the cancer cells, or they will suppress the induction of the cancer cells. So, you can actually use the RB proteins or the P53 proteins, and you will also use the BRC1, 2 and 2. And then you also can use some of the genes which are important into the DNA repair genes. So, some of these genes can also be used for the gene therapy in the case of the cancer cells. What is the approach you can use? So, you can actually use the multiple strategies. So, in the strategy number 1, what you can do is you can do something so that there will be actually inhibition of the cancer cell progressions. And the aim of this is that you actually bring the cell cycle control within the cancer cells, and you know that the cancer cell does not follow the cell cycle control, and as a result of that they actually proliferate for the indefinite period of time. So, what if you bring the cell cycle control, what they will do is after a certain period of divisions, they will actually get into the phase where they will not divide, and then ultimately they are actually going to die. And then you can also use the therapeutic genes like the tumor suppressor gene like P53, RB proteins or BRCA1, and then you can also use the antisense oligonucleotide or ribozymes or the siRNA. So, basically you have the strategy number 1 where you are actually going to make the cancer cells susceptible for the normal physiology. So, because of that they will actually go through with the process of apoptosis, they will go through with the process of death. Then in the strategy number 2, what you can do is you can actually deliver a suicidal gene into a cancer cells, right. So, what you can do is you can actually induce the cytotoxicity into the cancer cell with the help of a suicidal gene expressing cells, and this is a very novel strategy where you are actually inducing the death into the cancer cell. So, strategy number 1 is taking care of that cell, the cancer cell should not be able to survive or should not be able to survive for multiple divisions. In the strategy number 2, you are actually expressing the particular enzyme which will induce the death, and you can actually be having the gene activating the cytotoxic products. One example is where you can actually use the like for example, you can use the cytosine deaminase, and that will actually going to convert the 5-fluorocytosine into a 5-fluorouracil, and 5-fluorouracil is actually going to be the active drug, and it is actually going to disrupt the DNA or RNA synthesis, and as a result of that it is actually going to induce the cell death. And then we also have the another approaches, like you can actually have the tumor lysis by the recombinant viruses or the oncolytic viruses. You can also have the immunomodulations. Remember that the immune system is a very important factor to take care of the cancer progression, right. It's a immunosurveillance which is actually be responsible for the production of the cancer cells. So, when the cancer cells are being produced, they are being identified by the immune system, and that is how they are being removed from the circulation. But if the immunosurveillance system is weak, it is actually going to allow the proliferation of the cancer cells. So, if you are going to work with the immunomodulations, that also is going to take care of the cancer development or the elimination of the cancer. And then you can also have the anti-angiogenic and the anti-proteolytic gene therapy, where you are actually going to disrupt the ability of the cancer cells to form the new blood vessels, and as a result of that it will not get the nutrition and it will not get the way to spread to this site number 2 actually. So, this is all about the cancer. We have taken another example of SCID. So, SCID means the severe combined immunodeficiency. So, it is actually a immune system related disorder, where you are actually, it is a rare monogenetic disorder characterized by the defects in the development and function of the T lymphocytes. So, it is actually going to weak this person's ability to fight against the different types of infectious organisms. So, indirectly it affects the B cell lymphocytes, NK cell, and as they are dependent on the T cell. So, it is basically a defects of the T cell, but indirectly it is also going to affect the development of the B cell and as well as the NK cell. So, overall the SCID is going to induce the immunocompromised or immunodeficiency into a particular individual, and that as a result of that it will be more susceptible for the different types of diseases. So, for example, the children with SCID are known as the bubble boys, because some of them were actually able to survive for several years in plastic bubbles, in which the air coming inside was filtered and food and other objects were sterilized before introductions, right. So, the person with SCID is going to be extremely susceptible for the different types of diseases. And these are the symptoms of the SCID. So, it can be generally seen in the early days of life, right. So, immune system impaired and not capable of protecting the child. Therefore, children affected with the SCID are prone to a series of life threatening infections, including pneumonia, respiratory diseases, meningitis, ear infections, sinus infections, skin rashes, chronic cough and sepsis and all that. Now, what is the different types of SCID? So, you can actually have the adenosine deaminase deficiency SCID, you can have X-linked SCID, you can have the RAG1 and RAG2 deficiency SCID, and you can also have the IL-7R deficient SCIDs. And what is the strategies? So, you can actually harvest and do the gene corrections, you can actually do the conditioning, and you can also do the infusion of engraft, and that will actually going to cure the SCID in a particular person. So, this is another approach, right. This is another approach where you are actually going to do the germline gene therapy. So, so far what we have discussed about the somatic gene therapy, and we have taken 2 examples of the cancer and as well as the SCID. Now, we will take another example of the germline gene therapy. So, in the germline gene therapy, it is actually you are going to modify the germ cells, right. So, all the cells are being considered as the somatic cells, except the cells which are responsible for the production of the new babies, right. So, the germ cell means you are actually talking about the sperm and the ovum in the case of the humans, right. So, the germ cells, any kind of modification into the germ cells will actually be proliferate and will get into the offsprings like the new babies, right. So, the genetic modification of the germ cells such as sperms, eggs or early embryos would be a target site for the germline gene therapy. So, in this case what you do is you are actually, so for example, if you have the parents which are having the genetic disease, so what you can do is you can actually be able to do the manipulation. So, you can actually produce the baby, right, which is actually going to have a zygote, and this zygote is actually going to have the defective gene. Now, the embryo is also going to have the defective gene. So, what you do is you actually bring the embryo into the patty dish, right, and then you actually do the manipulations, and because of that this defective gene can be replaced with a new set of healthy genes, right. So, you are actually going to do the enucleation of the egg cells, right. So, you are actually going to remove the nucleus which is been formed from the parents, right, and then you will introduce the the new gene, or you will actually going to introduce the hepatic gene into the system, right, and as a result of that you are actually going to produce the improved zygote, and then you will put this improved zygote into the female, right, and you will allow them to grow, and ultimately you are actually going to have the healthy baby, right. So, embryos will be having the modified gene will actually give rise the healthy baby, and this healthy baby will actually going to be get rid of this particular disease for which you are going to do the modifications. So, simple example is the cystic fibrosis. So, cystic fibrosis is an autosomal recessive disorder. It occurs due to the mutation of the CFTR or cystic fibrosis transmembrane conductance gene, which codes for a 250 kilodalton transmembrane protein. The CFTR protein is activated by the cyclic AMP and act as a chloride ion channel, mediating the secretion of these ions outside the cell. And the CF is caused by the deletion of nucleotide triplets coding for the phenylalanine at the position of 508. Absence or the malfunctioning of CFTR mostly affects the respiratory and gastrointestinal tract by producing the very thick mucus, which rapidly colonized by the pathogenic organism, causing the chronic airway inflammation. So, this is cystic fibrosis occurs because there is a mutation of the CFTR protein, and that CFTR protein is actually not is allowing the proliferation of the pathogenic organisms, and that actually causes the infection of the air pathway. CFTR protein is a chloride ion channel involved in creating mucus, right, and digestive juices and sweat belong to the ABC superfamily and transport the chloride ion across the cell membrane, regulate the movement of water and sodium, that keep mucus thick and slippery, especially in the lungs, pancreas, sweat glands, intestine and the reproductive tracts. Now, in the cystic fibrosis, mainly lungs are mostly affected as the thick mucus is being produced, and mucus can also block the enzyme released in the pancreas and other organs too, and that is how it is going to cause the sinus infection or the salty tasting skins. And what is the treatment? So, there is no permanent solution for the cystic fibrosis curing the disease in a like. So, keeping the airway patient and suppressing infection by the antibiotic, you can take the mucolytic agents, and you can also use the CFTR modulators, such as these are the drugs. So, this is what it is shown here, right. If there will be a CFTR which is closed and open, right, and it is actually going to allow the release of the chloride. And remember that when we were talking about the transport of the material, we have discussed in detail about how these kind of the transporters are working, right. So, they will be actually forming a closed circle and open circle like that, with the help of the ATP and all those kind of loops are binding to the inner side, right. So, what is the gene therapy for cystic fibrosis? So, what you can do is you can take the healthy copy of gene into the adenovirus vector into a airway epithelial cells. Then the gene is delivering using the adenovirus associated by less side effect, but it is also but effective is also low. And then you can use the non-viral gene therapy using the liposomes and the polymeric nanoparticles, or you can use the antisense oligonucleotide therapies. In recently, you can also use the TALEN or the CRISPR-Cas to actually also alter the gene sequences, and as a result of that, it is actually going to help in the cystic fibrosis gene therapy. So, viral vector mediated aerosol mediated AAV2 CFTR gene transfer into the lungs of the patient with the cystic fibrosis was conducted by the Moss and al. And then you can also have the non-variable method. So, in a clinical trial where the compacted DNA nanoparticles were administered to the nasal mucosa of the cystic fibrosis subject to the check for the reconstitution of CFTR protein was conducted. Then the other disease is called as the dystrophin gene. So, muscular dystrophy is the X-linked disease caused by the mutation of the dystrophin gene, which is located on the short arm of the X chromosome. And the dystrophin is a very long filamentous protein located in the cytoplasmic site of the plasma membrane. Protein is composed of the 4 domains, see N-terminal domain, central rod domain, cysteine-rich domain and the C-terminal domain. Dystrophin mutation leads to the premature protein truncation, usually leads to the severe muscular dystrophy. If deletion in the central rod system, but the N and C terminal regions are intact, then the milder phenotype, that is the Becker muscular dystrophy. What is the gene therapy into the muscular dystrophy? So, dystrophin protein act like a shock absorbers, protecting the muscle fiber from the damage during the contractions. Without the functional dystrophin, muscle cells become fragile and are easily damaged, leading to the chronic inflammation and the replacement of the muscle tissue with the scars and fat tissue. It includes the delay of motor neuron, which independently affect sitting and standing abilities. So, muscular dystrophy is diagnosed by the level of creatine phosphate kinase, and which is involved into the muscle degenerations. Muscular tissue biopsy electromyographies can also be used for the confirmations, and through there is no cure. So, there is no cure for the muscular dystrophy, but few strategies are being used to delay the process, like for example, use of medicines like the prednisone and the derivatives of prednisone with the cyclosporine delays the muscle degeneration, and then you can use the monoclonal antibodies against the myostatin can also improve the muscle functions. What is the gene therapy in DMD? So, the probable gene for the GMD can be introduced the dystrophin cDNA via the adenovirus or truncated cDNA for the mini dystrophin production into the myofiber. We can introduce the whole cDNA of dystrophin gene in the plasmin and inject it into the bloodstream. We can suppress the premature termination of the dystrophin, or you can use the exon escaping use of ASO pairing, which masks the normal splicing signal on the dystrophin P mRNA with determining the exclusion of the pathogenic exon from the mature messenger RNA. All the processes are going on to the clinical trials. So, what are the steps in the gene therapy? So, you take the adenovirus associated virus, right, carrying the microdystrophin gene is infused into the patient's bloodstream. Once inside the muscle cell, the virus releases the microdystrophin gene. The cell's machinery then uses this new genetic print to bring the producing the microdystrophin proteins. The newly produced microdystrophin protein integrate into the muscle cell membrane, restoring the production and slowing the cycle of muscle degenerations. Then we also have an example of the hemophilia, one of the classical genetic disease where the blood clotting is a problem, right. And we discussed that in detail when we were talking about the genetic diseases, right. And we said that the hemophilia is a X-linked recessive disorder which body fails to form the blood clot, a process necessary to stop the bleeding. So, these are the cascade of reactions what is being responsible for the blood clotting, right. And there are many factors which are responsible for the blood clotting formations. And the different types of hemophilia, you have the hemophilia A, you have hemophilia B, you have hemophilia C, and they all are having the similar kind of phenotype, that they will be having a defects into the blood clotting. So, in the hemophilia 1 there will be a deficiency of factor 8, in the hemophilia B you have a deficiency of factor 9, and whereas hemophilia C there will be a lack of factor 11, but it also has autosomal recessive in nature. Now, what is the treatment of hemophilia? So, the treatment is the replacement of the clotting factor producing via infusion. So, it may be like prophylactic or episodic, or you can use the monoclonal antibody like the this antibody, which is a monoclonal antibody that mimics the function of factor 8, and it is highly effective for the hemophilia A. And this all provide the temporary controlling the symptoms of disease, not the permanent cure. What is a gene therapy? What you can do in the hemophilia? So, gene therapy provides a permanent solution. Here an effective copy of gene is inserted into the liver cell, which cascade makes the essential clotting factor to prevent the hemophilia. So, you can actually produce a gene, right, and you can actually introduce that into the system, right, and that is how it is actually going to start producing that particular factor. Then we also have the different types of gene transfer methods. So, we have the viral vectors, we have a non-viral vectors. So, within the viral vector you can have the choice of using the retroviruses, adenoviruses or adeno-associated viruses. You can use the herpes simplex virus, you can use the vaccinia virus or you can use the baculovirus. Whereas, in the non-viral vectors you can use the physical method or you can use the chemical method. So, physical method you can use the electroporations, micro injections or the gene gun. These methods you are going to use depending upon the type of organisms, type of target tissue, type of the target site. And within the chemical method you can use the liposome methods, you can use the calcium phosphate method or you can use the polymers. And then we also have the ex vivo approach. So, cells are genetically modified outside the body and then reintroduced into the patient. So, one of the classical example is the CAR T cells, right. So, you can actually isolate the defective genes from the patients, then you do the genetic modifications, then you are going to do the cell expansion and quality control, so that you will know that these cells are good and they will be in a high number, and then you are going to put these cells back into the patient, and that is how you are actually going to get the cure for that particular disease. So, what you are going to do is you are going to isolate the cells from the patient, you are going to do the in vitro culturing, and while you are doing the culturing you are actually going to replace the defective gene with the correct gene or the healthy genes, and then you are going to do the proliferation and then you will put them back into the system, right. For example, if there will be a problem in insulin production, so you can actually take out the beta cells, you can actually correct the beta cells, you put them beta cells back, right. So, for example, this is the example of CAR T cell therapy, right. So, what you can do is you can actually correct the T cells, you can collect the, so this is the patient, right, and you can actually be able to collect the T cells from the patients, then you are going to do the genetic alteration of these T cells. So, you will actually train them so that they can be able to cure or they can be able to identify the cancer cells and they will be able to destroy the cancer cells. And then you are actually going to produce the CAR T cells and they will express the different types of receptors and antigens for identifying the cancer cells. And then you are actually doing the conditioning chemotherapy, you will do the CAR T cell infusion into the patient back. And then once you infuse the CAR T cell into the patients, these CAR T cells are, since they already been having the recombinant receptor expressing on their cell surface, they will actually going to attack onto the cancer cells, and that is how they are actually going to eliminate the cancer cells. This is another examples of where you are actually going to take a therapeutic gene, then you insert that therapeutic gene into a suitable carrier. So, in this case, for example, you can use the adenovirus or the retrovirus or lentivirus, and then you will enter that into a patient, and then the patient is going to integrate that particular gene into its genome, and that is how the patient is going to start producing that defective gene, or they will start producing that particular protein, and that is how it is actually going to help to overcome the particular disease. And then we also have the gene modification. So, you can actually have the gene silencing or gene knockout, you can have the epigenetic modifications or the gene knock-in. So, in the gene silencing, what you do is you are going to do the siRNA or miRNA or the RNA, and that will inhibit the gene expression. So, basically it is actually going to suppress the production of that particular gene or the production of that particular protein, so that it will actually going to not cause any kind harmful effects. So, in the gene silencing is the regulation of the gene expressing without altering the DNA sequences. So, it is actually going to be temporary solution. So, it is only going to be until that particular transcript is available in that particular cell. And then we also have the RNA. So, RNA is another way of doing the gene silencing only. It works by introducing a small double stranded piece of the RNA which is called siRNA, and the siRNA enters the cell and loaded onto the protein called RSSE, and RSSE unwind the siRNA and uses one strand as a guide, and the RSSE guided complex patrol the cell, find the target messenger RNA and perfectly matches the guide and making it for the destructions. So, basically whether you are using the siRNA, whether you are using the gene alterations, and whether you are using the the knockouts and all that, it is actually going to only suppress the production of the particular protein from the particular RNA. Then we also have the antisense RNA techniques, that also is going to the same. It is actually going to reduce the production of that particular protein in that particular cell, and as a result of that it is actually not going to alter the genome of that particular cell. It is only going to change the transcriptome, and it is going to reduce the amount of RNA what is available for the protein productions. And then what is the advantage of gene silencing? So, it is reversible, it is highly specific, and it avoids the DNA risk, right. When you do the gene therapy, when you do the genetic manipulations, when you do the genetic mutations, right, you are actually changing a particular organism at a very gross level, right. So, that sometime may have the adverse effects, that sometime may have the different types of effects which probably you may not be able to know at this moment, but it may be available after like 3 generations or 4 generations. You probably will know that the target site is being modified, but off target site also could be modified, which probably you will not be able to know at that point. And as a result of that, many people think that if you cure the disease in a more safer ways, and the more safer way is that you just reduce the production of that particular protein by the gene silencing, and and because these processes are reversible, they are very specific, and they does not allow the alteration of the genome, they are probably will be much safer compared to the processes which are responsible for the genetic alterations. So, gene modification types, you can actually have either the addition of the gene, or you can actually have the gene editing and so on. So, in the gene editing you can use the zinc finger nuclease, you can actually use the TALENs, you can use the CRISPR-Cas. So, conclusions, gene therapy is no longer a question if, when, how, for ability to precisely rewrite the code of continuous to remove. It holds the ultimate promise of not just treating, but truly curing the most challenging human diseases. So, gene therapy is go beyond the rare diseases, development of more efficient delivery systems and the personalized medicines. So, in this particular lecture we discuss about the gene therapy, we discuss about the different aspects of the gene therapy, and we discussed that the gene therapy could be of 2 types. It could be the somatic gene therapy or the germline gene therapy. Somatic gene therapy is where you are treating a particular individual for a particular type of diseases, and it will not be inheritable. So, it will not go into the offsprings. And apart from that, you also can have the germline gene therapy, where you are actually going to do the treatment into either the germ cells such as sperms or the ovum, or you will actually do the alteration into the embryos, and as a result of that it is actually going to cure that particular genetic defect in that particular family, and as a result of that the subsequent generations are also not going to face the similar kind of diseases. Apart from that, we also taken an examples of the somatic gene therapy, we have taken an examples of the diseases from the germline gene therapy as well. So, we have taken an examples, we discussed the examples of the hemophilia, we discussed the example of SCID, and we also discussed about the muscular dystrophy. So, with this brief discussion about the gene therapy, we would like to conclude our lecture here. In our subsequent lecture, we will discuss some more aspects related to this particular course. Thank you.